Hey there, biologists! In this set of notes, we are going to be reviewing and going further in depth into the process of protein synthesis, also known as the central dogma of biology. Throughout the set of notes, I'm going to periodically interrupt it with a really amazing animation that walks you through this in a really good detailed visual about how this is actually looking inside of a cell. So we're going to start with a brief clip of an animation.
Transcription and translation are the processes that all living things use to read the information in DNA and build proteins. Each gene in a cell's DNA codes for a specific protein. During transcription, cells make a copy of a gene out of mRNA. During translation, they use that mRNA copy as a blueprint to build proteins out of amino acids. These proteins perform the tasks that make living things function. The flow of genetic information from DNA to mRNA to protein is central to life.
So as the video mentioned, this process of creating proteins is central to life. Without proteins, we would not be living organisms because they perform all the jobs inside of our cells. So this is why this series of steps is referred to as the central dogma, or central core rules or principles that govern how life works in biology. So the central dogma is two major steps and involves three molecules. We start with DNA, and in the process of transcription we make mRNA, and then mRNA is used to make a polypeptide, which is another word for protein or amino acid chain. When does protein synthesis occur? Well, protein synthesis is happening all over your body in every single cell, making hundreds of different types of proteins depending on what that cell specifically needs. So when a cell needs a specific protein to do a specific job, the specific gene or section of DNA will be expressed, in other words, turned on like a light switch, and protein synthesis will occur. So one example, we've talked multiple times in our course so far about the lactase enzyme and how lactase breaks down lactose milk sugar. So say, for example, you just drink a glass of milk, you have milk sugar floating around in your stomach, your digestive system cells will create that lactase enzyme, which will then break down the lactose milk sugar. There's some important background terms that you need to be very familiar with in order to keep up with this set of notes. Remember that a protein is a molecule that performs a job in a cell. An amino acid is a building block of the protein. Gene is a section of DNA that codes for a specific protein. RNA is a single-stranded nucleic acid that complements the double-stranded nucleic acid called DNA. And ribosome is the cell organelle that builds proteins. So let's start with the first step and go more in depth. Let's talk a little bit about transcription. The word transcription comes from its prefix transcript, which means copy. So a gene, or DNA, is used to make a temporary copy of that code in the form of messenger RNA, which is mRNA for short. In eukaryotic cells, this process of transcription occurs where the DNA is, which is in the nucleus of your cells. Whereas for bacterial prokaryotic cells, this process takes place in the cytoplasm because they do not have a nucleus. So again, to recap, we start with our double-stranded DNA molecule. One strand of that DNA molecule will be used to create mRNA, which is a small temporary copy of part of that original code. And this process is called transcription. Here's an image that shows transcription in more detail. We can see that we have our original DNA template that's being opened by RNA polymerase. We've learned about polymerase and how polymerase builds a polymer, and in this case we're building RNA. And so one strand of this double helix DNA is used to make a temporary copy of that code that codes for the specific protein. Before I move on to translation, I want to show a brief animation of this process and what it would look like step by step inside of a cell.
Inside the nucleus, RNA polymerase attaches to a gene. It separates the two complementary DNA strands, exposing the gene's unique sequence of bases. When the end of the gene is reached, transcription is complete. The new strand of mRNA passes out of the nucleus and into the cytoplasm, where translation.
So that animation showed transcription in a nutshell, again using DNA to make mRNA. And what's really important is that you understand the base pair rule here. mRNA does not have T. So anytime you would have written a T when you're building an mRNA strand, you instead replace the T for U, which stands for the nucleic acid uracil. Excuse me, nucleotide of uracil. All right, let's go in depth into the process of translation. The word translate means to change the language. So in this step, we are taking the mRNA, which is a nucleic acid language, and we are translating it into the language of proteins, which is amino acids. So the mRNA will travel to the cytoplasm and it will bind to a ribosome. The ribosome will then translate that mRNA into the amino acid language of a protein. So again, in a nutshell, mRNA, cytoplasm, polypeptide, which is made up of amino acids, and this process is translation. The ribosome is critical in this process. Let's take a look at this process in a little bit more depth. So the mRNA sequence is read by the ribosome in 3-letter words called codons. Each codon, or 3-letter sequence of bases, codes for one specific amino acid. There are 64 possible codons, or 64 possible 3-letter combinations of AUGC. And these 64 possible codons code for 20 total amino acids. So therefore, you should be able to understand that each amino acid might have anywhere between 1 to 6 possible codes that code for it. We can understand how this works by using a codon chart. So say, for example, I extracted an mRNA molecule out of a cell, and I wanted to know what specific type of protein it codes for. What is the sequence of amino acids for that protein? Well, I can use a complex series of steps to read the A's, U's, G's, and C's of that mRNA in order to understand the amino acids. And the ribosome will do this process as well. It will begin by building an amino acid sequence when it encounters the first AUG sequence. AUG codes for the start codon, and therefore every amino acid of every protein starts with methionine, or MET for short. And in order to use this codon chart, again, you start with your 3 letters of mRNA. The first letter is the biggest letter in the middle. So we have A, U, G, and that stands for MET, or methionine. All right, let's do some practice. Here is your mRNA sequence. Translate the mRNA bases into the amino acid sequences using this codon chart. Pause the video to do this. All right, let's check your work. Again, AUG codes for the start codon methionine. CCG codes for proline, so CCG. You can also see that CCA, CCC, and CCU also code for proline. We will talk a little bit about the importance of that coming up when we get to genetic mutations. UGA is UGA, and this codes for stop codon. Stop codon does not actually translate to a specific amino acid. It basically just signals the ribosome that this process is done and over. So here are your answers. So therefore, this protein is 2 amino acids long. You have methionine bound to proline, and that is your total amino acid sequence or protein. So how exactly do the amino acids get to the ribosome in the correct order? This is where another really important type of RNA comes into play. Transfer RNA is shortened into tRNA, and it is a type of RNA that's floating around in the cytoplasm. tRNA is, like I mentioned, our type of RNA, so it's a single-stranded RNA sequence that is bound to itself in this kind of conformation, and it actually looks like a T shape. If we were to look at that in its actual protein or in its 3-dimensional folded shape, it would look more complex like this. And we usually see it simplified in a number of different ways. And it always will be attached to a amino acid if it's being used in the process of building the protein. So the job of the transfer RNA is to transfer or carry amino acids to the ribosome. Every tRNA has an anticodon. So this anticodon, remember the prefix anti means opposite. So the anticodon is 3 bases of RNA on the tRNA that correspond to the mRNA codon. And the anticodon will determine the type of amino acid that it carries, and therefore when it will be carried to the ribosome to be used to build a protein. So let's take a look at this process in more depth, starting right here. So tRNA molecules carry amino acids to add to the polypeptide chain. So here's a tRNA floating around in the cytoplasm. This specific tRNA has the anticodon AGU, which would code for the mRNA codon UCA. Okay? And if you were to translate UCA into the amino acid language, you'd actually see that it does confirm that it codes for serine as the amino acid. So the tRNA carries the specific amino acid based on the sequence. And this all has to do with a complex series of chemical attractions between the ribosome, the mRNA, and these tRNA molecules so that it attracts the correct tRNA and therefore the correct amino acid. So our next mRNA sequence that's going to be read by our ribosome is GGU, which therefore codes for CCA as the anticodon, and GGU codes for glycine. So tRNA carries its amino acid to the ribosome and it will drop off its amino acid. A type of bond called a peptide bond will form between all of the amino acids in this chain, and they will bond together to form what we call the polypeptide, many peptide bonds. And so this will form our protein. Once the tRNA drops off its cargo, its amino acid, it will disconnect and it will go back to the cytoplasm, where it will bind to its correct amino acid that's floating around again, and it can be recycled and reused for this process all over again. So the conclusion of translation. When the ribosome reads one of 3 possible stop codons, the mRNA, the ribosome, and the amino acid sequence will disconnect from each other. All of this can then be reused for different parts of this process again. The amino acid chain will then fold into its completed protein and travel to its end destination. So here's our completed sequence of amino acids. Through a complex series of chemical attractions and bonds, they will form this 3-dimensional shape, which then forms our final protein. And in the example we talked about earlier, we were talking about making lactase. So in the cells of your small intestine, they would be doing this process of creating lactase, which would then gets released out of the cells into the space in the digestive system, which then breaks down lactose. When a protein is no longer needed, the mRNA transcript will be degraded or broken down into its individual nucleotides, and the protein will be degraded into its individual amino acid. And what's super cool about this process is that all of those nucleotides and all of those amino acids floating around can then be used to build all new mRNA sequences or all new amino acid sequences all over again. So everything is recycled and reused in your cells. Let's do a pause in the notes and let's go to the animation to see what this looks like in a cell.
And into the cytoplasm where translation takes place. The instructions in an mRNA strand are read in groups of 3 bases called codons. Each codon must be translated, one after another, in order to build a functioning new protein. The key structures in that process are the ribosome and transfer RNA, tRNA. Each tRNA molecule has its own anticodon and a matching amino acid, the building blocks of proteins. To begin translation, a tRNA molecule coded for the amino acid methionine docks with a ribosome. The ribosome attaches to the end of the mRNA strand. It slides along until it finds a start codon, AUG. Through complementary base pairing, the start codon pairs with the anticodon on the tRNA molecule, signaling the beginning of the protein instructions. The methionine molecule is now the first amino acid in the new protein. The ribosome shifts to read the next codon. Each codon is recognized by one of 20 different tRNA molecules. Each type of tRNA carries a different amino acid. The ribosome continues moving along the mRNA strand, one codon at a time, matching it to a complementary tRNA. It adds each amino acid to the growing protein in just the right order. The sequence of amino acids determines the final shape of the protein and the function that it will carry out. Give translation a try.
So my mRNA codon is GUA, so the opposite of this would be CAU. AUU is complementary to UAA. AUU, UAA. CCA, GGU, which codes for proline, the amino acid. So you can see that these amino acids are being brought to the ribosome and are creating this overall chain. GCU, CGA. Alanine. UCG, AGC. Serine, or SER for short. AUU, UAA. Isoleucine. GAU, CUA. Aspartic acid. GGC, CCG. Glycine. AUA, UAU. Isoleucine. CGC is the same as GCG. Arginine.
When the ribosome reaches a stop codon, it detaches from the mRNA strand. The chain of amino acids folds into its final form and the new protein is complete. This protein, DNA helicase, will now slide between two complementary strands of DNA, temporarily separating them so that they can be copied. The incredible processes of transcription and translation enable cells to carry out all of the functions necessary for life.
All right, so in this set of notes, we went in depth into the process of transcription and translation. And make sure you're sending me messages if you have questions. Connect with me during Zoom class, join me for office hours, and let me know if you have anything you need help with. Thank you for watching and have a wonderful rest of your day.